Hey there. Imagine you're at a busy party. One juicy rumor starts in the corner and, person by person, it sweeps across the room. Parkinson's disease works a lot like that rumor. The trouble doesn't burst onto the scene. It hops from one brain cell to the next. Today we're talking about a protein that seems to play the role of the gossip carrier, and what happens when scientists shut it up. Here's the big idea. Instead of chasing every single bit of misfolded protein, researchers asked, what if we block the delivery service that moves the trouble around? The delivery service, in this case, is a protein called GPNMB. Cut off that courier, and the rumor, the toxic form of alpha-synuclein, can't spread as easily. First, let's set the stage. 1. The damage starts. Misfolded alpha-synuclein shows up. 2. Immune cells called microglia sense the trouble. 3. GPNMB helps package that bad protein into tiny particles, which then jump to healthy neurons. If any one of those steps stalls, the chain reaction slows down. Scientists zeroed in on step 3, GPNMB, because it kept flashing red lights in every experiment they ran. Now, how did they spot it? Picture a slow-motion replay. Researchers used cultured cells and special mice that develop Lewy body-like clumps along predictable brain routes. They pulled spinal fluid, mapped genes, and watched which molecules light up when the spread is at its worst. Over and over, GPNMB was front and center. But showing up isn't enough. You need proof it matters. So they painted brain slices. Under the microscope, GPNMB glowed on stressed microglia and at the exact handshake points with neighboring neurons. High-resolution images even caught GPNMB gripping little cargo vesicles loaded with toxic alpha-synuclein. It looked less like a bystander and more like a loading dock attendant. Here comes the critical test. Scientists deleted the GPNMB gene in microglia. Guess what? The misfolded seeds were still there, but they moved in slow motion. Fewer vesicles formed. Neurons picked them up reluctantly. Then they tried a clever twist. They re-added GPNMB, but with a mutation that can't grab cargo. Spread stayed sluggish. Bottom line, the gripping action itself is essential. Think of it the way crystallographers throw a tiny wedge into a growing crystal lattice. The basic building blocks remain, but the whole structure can't extend. Knocking out, or even just hobbling, GPNMB adds that wedge at the cell-to-cell interface. The bad proteins lose their Uber driver. Let that sink in for a second. Okay, so where does this lead? Drug designers are now sculpting antibodies that stick to the exact surfaces of GPNMB involved in vesicle loading, while sparing parts the rest of the body still needs. Human stem cell-derived neurons and mini-brains, yes, organoids, are helping dial in the sweet spot. Block enough to slow spread, not so much that microglia stop cleaning up everyday debris. Here's a handy analogy. In classic enzyme kinetics, a non-competitive inhibitor clamps onto a regulatory site and turns the reaction volume down without trashing the enzyme. These GPNMB targeting antibodies aim for the same finesse: more dimmer switch, less sledgehammer. A second analogy comes from virology. Neutralizing antibodies don't blow up viruses. They cap entry points so each viral particle has a lower chance of infecting a new cell. Do that to toxic alpha-synuclein particles, and, over years, the expansion curve of brain damage could flatten. Early lesions may still appear, but the wildfire doesn't rage. Why should you care? 3 reasons. First, the goal line moves from just easing symptoms to actually slowing the march of damage across the brain. Huge shift. Second, it opens the door to combo therapy. One drug blocks spread, another tackles inflammation, a third hits genetic risk, much like multi-drug cocktails revolutionized HIV care. Third, and please keep this in mind, this work is still in models. No prescription is coming from your neurologist tomorrow. But understanding the mechanism is how tomorrow's treatments get born. So, where are we right now? GPNMB is no longer an obscure glycoprotein. It's a clearly defined target with adjustable knobs, not an on-off switch. Researchers are already comparing notes across Parkinson's, Alzheimer's, even ALS, because harmful protein spread pops up in all 3. Before we wrap, let's zoom out and make this personal. If you had 10 minutes with the team blocking alpha-synuclein spread, what would you ask about risks, timelines, or trial access? Could you bring those questions to your next support group meeting or doctor's visit? Also, knowing how pathology hops cell to cell, is there a research registry you might join or a clinical trial newsletter you could sign up for today? Finally, does realizing scientists can target a single helper protein shift how you plan your own care, medically, financially, emotionally? Maybe that nudge is all you need to update a care plan or double down on that exercise routine you've been eyeing. Exceptional recap. So, here's what we covered today. Parkinson's damage spreads like a rumor from neuron to neuron, helped along by the protein GPNMB. Delete or hobble GPNMB, and the toxic cargo still exists but moves far more slowly. Think wedges in a crystal lattice or a dimmer switch on an enzyme. Drug developers are crafting antibodies that clamp GPNMB's loading dock while sparing its other chores, aiming for finesse over force. This shift targets the root spread of disease, invites combo therapies, and echoes strategies used in virology and enzymology. We're early in the journey, mouse brains and cell dishes, not pharmacies, but every breakthrough starts exactly here. Remember, every so-called failed trial can redirect researchers to the real culprit, just like studies on protein spread led straight to GPNMB. Progress often hides in plain sight. So the next time you hear about a setback, ask yourself, is this the clue that sparks the next leap forward? Because that's how science works, one rumor stopped, one cell at a time. Thanks for listening. Take a breath, share what you learned, and stay curious. Until next time.